Good afternoon. So today we're talking about harvest aids, or actually, more accurately, I should have labeled this PGR. So we're talking about PGRs today, plant growth regulators. And again, this is kind of a small chapter, so we'll just kind of get right to it. These are the 5 essential plant growth hormones that I want you guys to know. So basically, you have auxins, cytokinins, gibberellic acid, ethylene, and abscisic acid. So these are the 5 essential plant growth hormones. In ag, you should all kind of have these memorized. So I'm going to require you for the exam to know these definitions. In another class, they'll probably have you know the 16 essential plant nutrients, but like I said, that's a different class. But just know that it's good to know these 5 as well as the essential plant nutrients. So auxins, so also known as IBA, are produced in all new tissues. It involves cell division, moves food to new tissues, and stimulates root growth. So basically, these hormones stimulate uh, plant growth and tell the plant what to do. It's like it's already in the genetics of the plant to do certain functions. These hormones actually turn on those functions and they tell the plant, okay, now it's time for you to do this, now it's time for you to do that. They're just signals is all. So these naturally occur in the plant at all times. We can artificially apply more of them to get the plant to do what we want at a certain time, but these occur naturally. The next one, cytokinins, that's produced in roots. Cell division controls hormone cycle, which is the aging of plants. So basically, it's telling it what to do, when to go reproductive mode, and all of that, because as described in the last class I taught to you guys, there's a difference, a big difference between vegetative and reproductive. So this helps kind of control that. Gibberellic acid, so that is produced in all plant parts, especially seeds, and it increases the size of the cell. So basically, you are expanding the cell wall. So it's not just it's not that you are adding new cells, making the plant bigger. You're just taking the current cells and just making those bigger. Same number of cells, they're just expanded larger. So then ethylene, so that helps with fruit ripening, and increases the movement of auxins. It's the first line of defense against stress and disease. Ripening, that's the thing I want you to know right there. So that's the most important part with this. So in your notes, make sure you indicate that it controls ripening, for sure. And abscisic acid, so that moves sugar out of the leaves and terminates cell life, which causes grain seed to go dormant or dry down. So basically, that's it there. It causes seed dormancy. So seed dormancy for abscisic acid, ethylene controls ripening, gibberellic acid increases the cell wall size, cytokinins control the aging of plants, and auxins help with new root and shoot growth. So put that in your notes. That's what I want you to know for the test. That's a big part of this chapter. So each of these we can control artificially, as I mentioned. Let's see here. So when it comes to auxins, one type of auxin is IBA, or indole 3 butyric acid. So this is what we use as a rooting gel. Since it encourages root growth, you can actually buy this stuff and dip a cutting, what we call it as a cutting is just a piece of a plant stem that we remove, that we want to from a plant that we want to clone, and then we dip it in this gel and get it to grow roots. So you dip it in the gel, and then you put it in some potting soil. I've done it a lot at home. It's kind of a hobby of mine. If you go see a tree or a plant that you really like, you can actually clone it this way. I've done it a lot with grapes. So, if you guys are interested, you can look into that. But this is a big use for it. It's used a lot in nurseries with tree crops and grapes and stuff like that. So just keep that in mind. It's a good way to clone a plant, because when you grow a plant from seed, you have a mother and a father, and sometimes those genetics don't quite give you what you want. To put it in, you know, kind of a funny term to say is you don't know who the father is, right? So like if you have a tree and that tree is receiving pollen, you don't know where that came from. It could have come from a weird oak tree. Let's say you're trying to breed oak trees, you're trying to plant the seeds of an oak tree. Um, it's of course going to come from another oak tree, so it's not half tomato or something weird, but um, you don't know what the father looked like. You don't know what that male oak tree, I call it male, but the the tree that gave you the pollen, you don't know if that was a desirable tree or not. So this is a good way to clone. If you have a tree you really like, you like its attributes, the size of the leaves and whatnot, this is a good way to copy it. So gibberellic acid. So remember, this is the one that expands the cell walls, just makes the cell walls bigger. It's used a lot on grapes. So here's a great example. On the left, that's no gibberellic acid. On the right has gibberellic acid. So same level of sugar in both of those fruits there, yet the one on this side of the screen here has the gibberellic acid, which just expands the cell walls, and all it has is it has more water in it. Gives it more. Um, it just gives it more water, bigger cells. That's literally it. No more sugar. So if you were to eat one of these smaller grapes, you might actually think that it tastes sweeter, because you're getting it more concentrated. And so pretty much all grapes you eat in the store have been sprayed with gibberellic acid. And I can see it now. Everybody who hears this always complains. They go, "Oh, but it's not natural." It is natural. It's something that the plant is already producing. We're just giving it a little bit more of that. So it's not dangerous in any way. Um, all it is cell wall expansion. Here's another usage. We're kind of going down the train here. I'm trying to cover examples that you would see or are relevant to this area. Obviously, we don't really grow any grapes around Yuma, but um, you do see it in the grocery store, and it's a great example of gibberellic acid. But here's a lemon that's not quite ripe. You see, it's got some green on it. So a lot of lemons have to be picked green because they're trying to hit a certain market time. The desert lemons, you get more money from them because we can ripen them quicker. They get to size quicker than the ones in Florida. So if you can get your Yuma lemons out first in like September, then you're in good shape. If you wait, then the market price won't be that good. So a lot of the time we'll pick the lemons, ready or not. They're a certain size, they have everything we need them inside, but they're still green. They're not yellow yet. They're not quite ripe. So even though the size is good, so we pick them, and the yellow ones go straight to the box and they get shipped to a store near you, but the green ones get sorted out and they go to an ethylene chamber where it gets flooded with gas and it actually turns them yellow. Like I said, perfectly natural. This is what fruits have already in large amounts. Um, you can do this at home. Just if you have a tomato or a fruit or a lemon that isn't ripe, you put it in a brown paper bag, and the ethylene that fruit naturally gives off will actually ripen it for you. So that's a good example of it. They need it done quicker, and they need it done more uniform, so they put it in a chamber with ethylene pumped into it to do it quicker, but you can do it at home just as easy. So PGRs, so you can also use them as pesticides. One good example is a product called 2,4-D, and when you take the PCA test, you'll notice that it's actually it's on the test. They'll want you. There's quite a few questions about it, especially the Arizona test. And what it does is it gives the plant too much auxin, which, if you remember, auxin is the plant growth hormone that stimulates shoot growth. So basically, what happens is the plant just grows itself to death. It gets kind of twisted look. It grows too quickly and dies. And it targets broadleafs, but not grasses. Remember that distinction when we talked about weeds? So that's the deal there. Grows itself to death, and it saves the grass. So it's a selective herbicide, to use our terms here. Defoliants. So now for the rest of this class, we're going to be talking about defoliants, and this is primarily used in cotton. It's used in other things, but I want to focus on cotton because that's the main reason why you have this license here in Yuma, Arizona. So having a PGR license could help you to apply any of the products I just mentioned. But 2,4-D is kind of a tricky one. It is a PGR, but that's a weed license. That's you need to be a weeds PCA to apply that because of what your target is. Now, if your target is to defoliate a crop, then you'll need this PGR license. So defoliation of a crop, basically, what is it? It basically makes the leaves abscise. The leaves drop, kind of like how a tree in the winter loses its leaves. It's the same exact thing. The plant doesn't die. It just drops its leaves and prepares for the winter time. So basically what we're doing is we're forcing cotton to do that. And it basically kills the plant foliage. Excuse me. That's a defoliant. A desiccant kills the plant foliage. It makes the plant die. So if I ask you on the test, what's the difference between a defoliant and a desiccant? Defoliant makes the leaves drop without killing the plant. Desiccant kills the plant. So there you go there. Another term here, senescence, is the process of aging in plants. So basically, chlorophyll degradation causes the autumn leaf color you see when like a really nice oak tree turns orange in the fall. That's what it is. It's a chlorophyll degradation. And there's a bunch of chemistry there that we don't really need to get into. And then abscission, already mentioned. So when a plant drops its leaves over its fruit. So just a few terms there before we get into this. So why do we want to do this? Why would we want to use these products? Well, basically, we can use them to cure seed crops, crop residue sanitation, make the harvest easier, and make a harvest earlier than normal. So basically, when it comes to cotton, we need to get our stuff harvested before the produce season starts. Now you can grow cotton and it'll just naturally sort of die off once it or lose its leaves anyway once the cotton is ready to pick. But we don't have time to wait for that because we're planting in the spring and then we're going all the way till September, and then we want to be done because we want to plant produce afterwards. So we do not want to kill the cotton plant. We want it to remain alive so it can accelerate the natural process of senescence where it loses its leaves naturally, so that we can pick the cotton instead of just flat killing it. So again, why would you want to do this? Well, let's say you're growing cotton in a cold weather region and you want to get your harvest in before the cold weather starts, before a frost happens, or to start produce season. That's why we do it here in Yuma, to begin the produce season, because produce is a much more high dollar crop. So that is the situation there. So how does defoliation take place? The cell walls are weakened in the abscission zone. And basically, a healthy leaf would remain attached to the plant indefinitely, and it'll only abscise if it becomes unhealthy or senescent. So unhealthy leaves drop and old leaves drop when the plant tells it to. So we want to be able to tell it to drop sooner. So you're kind of causing an unhealthy leaf by forcing it to drop. Um, Plant must be fully mature to apply this product, and temperature must not exceed 80 degrees during the day or 50 degrees at night. So we're pretty much okay to do this during the fall. And then it really gets into chemistry. I don't want to cover that too heavily. But just know that, humidity, wind, temperature, always check the label. Remember, that's always the answer in this class. Can I apply this product? I don't know. Check the label. See what the label says. The label is the law. So I'll probably ask that again. I know I asked it 3 times on the midterm, and I'll definitely be asking it on the next test. How do you know this? And the answer is always check the label. So which crops are commonly defoliated or desiccated? Cotton. And we do what we call a pick spray, allows for earlier harvest, reduces seed cotton moisture, reduces boll rot, enables mature bolls to open sooner. The bolls are these kind of round spheres where the cotton grows inside. Fewer cogs in the cotton picker and reduces infestation of insects. So the cotton effects on ginning, a cotton quality preservation, reduce trash that may interrupt the ginning process, reduce moisture, which reduces ginning. Um, basically, the plant conditions and second growth. When all healthy bolls have reached maturity, the plant will go back into a vegetative state of growth. Like I said, it goes on about that. I don't need you guys to know too much. Just basically what I've covered. Just take notes on what I've covered. And this lecture will remain posted on Canvas. This isn't a very complex sort of category here in the Yuma Valley. So I just wanted to show you guys this. This is the difference here between a treated section of cotton with a PGR versus untreated. The untreated is the green part. So we didn't put any PGR in there, no defoliants on there. So that's the difference there. The treated section has much more bolls of cotton open, no green leaves to clog up your cotton picker. You can run in there pretty easily. So that's kind of how it works. And then apparently you can use it on alfalfa. Fewer cogs in the combine. This is more for seed production. Reduces shattering. Shattering is when your seed pods open and it drops the seed to the floor, so it can reduce that as well. So this kind of stuff is only really used in the production of things that produce seeds. Cotton is technically a seed, or at least has seeds on the inside of it. So I suppose you could call it a fruit if you wanted to. So basically, you got to follow the rules on this. And remember, once again, the label is the law. They have some advisory rules on this, and then views and the toxicities, and always follow the label. So remember, the answer on this is follow the label. So that's about it. So I have for you guys a discussion to do, which includes reading an article and then talking about it, and then the quiz. So no lab for today, just the quiz and the discussion, and I will see you all next week. Thank you.